take it.
Thank you all very much. Good morning. As Kathy said, my name is Joshua Voss. I'm a coral reef ecologist and molecular ecologist. My background is in coral disease ecology to start with. Broadly, I'm interested in finding, discovering, and characterizing reef habitats throughout the Caribbean and worldwide so that we can understand and protect those habitats. Specifically, that also sometimes relates to investigations of threats to coral reefs, including coral disease, land-based sources of pollution, fishing impacts, boating impacts, etc. When I first came to Harbor Branch about 6 years ago, my entire scope of work was far afield, middle of the Gulf of Mexico, far off in the Bahamas, etc. And I was recruited by Jeff Beal from FWC to come and look at this local site because it was so interesting. So we've been working there since roughly 2010, and what we realized is a huge gap in response to the water quality issues that we have in the lagoon was that we had many, many different research groups looking at oysters, seagrass, fish, etc. within the lagoon system, and there was essentially no one quantifying any effects on the coral reef ecosystem itself offshore. So we were trying to fill that gap. My role at Harbor Branch is assistant research professor. I'm part of the Coral Reef Conservation Program within the Center for Marine Ecosystem Health. I'm also a PI on a NOAA Cooperative Institute for Ocean Exploration Research and Technology. So many of the techniques that we've developed globally, we've applied specifically to St. Lucie Reef. What I'm going to talk about today is research that's been funded in part by a state wildlife grant and partnership with FWC, as well as a donation from the River Branch Foundation, support from our state wildlife, or excuse me, our state specially licensed plates, and recently a Florida Sea Grant. And we've also had very great partnership from the park since St. Lucie Reef falls within St. Lucie Inlet State Park. So as a reminder, in terms of importance of coral reef ecosystems, they're very high centers of productivity. And when I say high productivity, that means they're taking sun energy and turning it into fixed carbon that we can eat up the food chain. So we don't have our fish if we don't have productivity going on at lower trophic levels. And as a result of that productivity, especially on coral reefs where we see lots of benthic or sessile organisms on the bottom, we tend to see lots of biodiversity as a result of habitat partitioning. So we have really high biodiversity on coral reefs relative to other places in the ocean. Coral reefs only occupy less than 0.2% of the ocean bottom, but more than 26% of known species in the ocean occur on coral reefs. So they're very disproportionate in their role of productivity and species diversity. They offer great coastal protection services for us in terms of ecosystem services, as well as provide vast tourism industry, potential pharmaceuticals, support fisheries, etc. There's been a varying range of estimates of the value of coral reefs, but generally accepted is somewhere between $80,000 and $240,000 per square mile per year is their estimated value. And if you look at the entire CEFTRI region, it's estimated to contribute about $3.4 billion annually to our region. And that's a combination of fishing, tourism. That includes things like the amount a tourist or diver might come to spend on a hotel and food when they come to our region. Unfortunately, we're seeing lots of areas where we see degradation, from things like you see on your left to things like you see on your right, and in many cases we have no idea why, and we want to understand how to reverse that trend. And so that's been the focus of most of the work I've done in relation to the CEFTRI region. So I'm hoping all of you have been to St. Lucie Reef before. If you have not, I very much encourage you to try to get out there. It's right out our doorstep, just out the inlet. And again, the techniques and approaches I'm going to talk about here are replicated at multiple marine sanctuaries throughout U.S. jurisdiction waters, as well as Belize and all throughout the Caribbean. So this is just a snapshot of this one location. But this one location is extremely unique because it's at the northern end of the Florida Reef Tract. It's the latitudinal limit for a number of species, such that if you go on the other side, the north side of St. Lucie Inlet, you see many species of corals drop out, many species of fish drop out of the population. So we know that this particular region is quite unique biologically and ecologically. has extremely high fish diversity for a coral reef. Over 250 species of fish have been documented. And then in the teens to 20s for sponges, hard corals, soft corals, etc. That hard coral number is really interesting because there's 24 that we've documented occurring at St. Lucie Reef. If you jump to the north side of the inlet, that number drops down to 5. So this particular reef is the northern limit for at least 19 species of corals along the Florida coast. To give you an idea of kind of the layout of this reef, here you can see that we have a series of northwest to southeast running ridges. Most of these are consolidated sediment or worm rock overlying with coral colonies. So in contrast to the lower Florida Keys, where the actual reef structure is built up of corals themselves, here most of our coral reefs are corals on a coral veneer on top of other kinds of consolidated sediments. You see the most worm rock up here around the inlet, and as you move down, we start to see larger colonies, such as this large brain coral that's almost 3 meters in diameter. And this brain coral and this mountainous star coral, Montastrae cavernosa, are the two that really dominate the ecology in terms of corals at this reef. They're both the largest and most numerous species of corals that we see at St. Lucie Reef. But we also see a lot of Oculina diffusa, as well as some Oculina vericosa and other species. This is an ivory tree coral. I bring this up primarily because this coral is the one that is apparently the most impacted by fisheries in this region. We very frequently find cast nets entangled on these branching Oculina corals every time we go out. We do see some cases of what we would call adverse health effects on corals at this reef, and so some of our initial investigations mapping this reef, getting a handle on the species that were present and where they were present, we found incidents of what's called coral bleaching. So corals are all cnidarians. Imagine taking a jellyfish and turning it upside down like an anemone, gluing it to the bottom, and having that anemone replicate, clone itself all over the surface of the coral. That's what makes up a coral colony. But that productivity is driven by the fact that they all have small algae living inside their tissues. So most corals are essentially algal farmers. Sunlight is providing all the energy for that coral, and they supplement it with just a little bit of feeding. If corals experience stress, and that can be stress from discharge water or thermal stress, both hot and cold, or UV stress, they can lose those algae. That symbiotic association can break down, and the corals bleach. So what you see is just bare coral or very pale coral tissue. If that condition occurs for just a short amount of time, corals can and do recover, but if those adverse conditions persist, ultimately the corals die. So it's not just the intensity of stress events, but the duration of stress events that can ultimately lead to potential coral death. So what we've done is essentially start to set up a network within this system with two major goals. The first is developing metrics to assess coral health before corals die. You asked before, or someone asked before, if we were just going to watch them until we don't see them anymore. And most monitoring programs are designed to assess the relative abundance and number of corals. But it does you very little good to say this many corals died and we don't know why. What we're trying to do is use advanced molecular techniques to diagnose stress within corals before they die, so that we can potentially reverse what's causing those stresses if it's within our scope and within our capability before those corals are lost. And I'll give you a couple of examples of that later on. Our second goal was to create a baseline of overall coral health and coral assemblages in this region in light of the comprehensive Everglades restoration plans that are in place currently, as well as potential additional measures that may be taking place at local levels. So we understand what's going on now, and we can track potential improvements to the system over time. I hope nothing more than to have definitive data that I could bring back to the comprehensive Everglades restoration team and say, we made this change. It was expensive, it was difficult, but we had this positive outcome occur on the coral reefs. I think that's very important. And as a reminder, this is St. Lucie Inlet State Park, and so we've worked hand in hand with the Park Service throughout this entire project. I'm not going to spend too much time on this because Curtis already spoke about it quite a bit. You all are quite familiar with our watershed structure here. and of course our very simple and easy to understand Lagoon operating system, which has a number of decision points within it. And if any of you have not yet, I encourage you to participate in the weekly calls where they discuss the regulatory system and make the decisions about these individual nodes within this decision support tool. And you'll come to understand that while we may view this system as very objective criteria for release, the calls themselves have a bit of subjectivity in them as well. If you call in and you're not a part of the team, you're welcome to listen in, but you cannot speak. So I encourage you to do that. And most of you are familiar with what happens during discharges here on St. Lucie Reef. We go from crystal clear water that happens on occasion to incidents of black water that can persist for days up to a month, month and a half has been the longest that we've documented to date. Typically, the delineation between offshore water and black water or discharge water is very distinct, like you can see here in this photo that Jackie Thurlow-Lippisch took, as well as some of these other images as well. And so what we've tried to do is target our sampling to occur over time at regular intervals, as well as to sample during these discharge events to look specifically at the effects of the discharge events. So we've established 4 different locations along the reef. So here's the same image I showed you before, just a different orientation. And at each of these locations, we have identified individual coral colonies, 5 of each of those 2 major species, the brain coral and the star coral, so that we could go back to them over time, look at their growth rates, look at their tissue levels, et cetera. And I'll give you more details on that shortly. In addition to the biological measurements that we're taking in these areas, we've also installed salinity and temperature loggers at each location, so we can actually track the pulse of reduced salinity water as it comes out over the reef. And then we also have loggers right there in the inlet. Also, as a reminder, this is a heavily used and very important fishery, particularly for kingfish, mullet, etc. So there are many occasions when we go out and there are upwards of 30 or 40 commercial boats in this area at any given time. So it certainly has a big economic impact. So in terms of tracking the salinity, here again is our 4 reef sites and the inlet. Those colors correspond directly to what you see here. And this is data from roughly this time last year. So you'll notice that, if you recall, we had an unusually wet January last year. Should have been the dry season. Instead, we had huge thunderstorms right in the middle of the first week of January. So we go from moderate salinity. Normal for this area is about 34 or 35 parts per thousand, with a range down to 30 being fairly normal. But right after those huge rain events, both in the inlet, here shown in orange, as well as at all of our sites within St. Lucie Reef and St. Lucie State Park, we see this dramatic drop in salinity that persisted for about 3 weeks and then returned almost to normal. So what we wanted to know is, can we relate these changes in salinity that we're seeing on the reef directly to either rain events or discharge events? And so if we compare it to the rainfall at the time, you'll notice that we did have 2 other rain events in November and early December here. And you see slight dips, but not the dramatic drop in salinity that are occurring on the reef. In other words, our system is resilient to these moderate rainfall inputs in terms of its ability to handle salinity. But when we had that huge event, which I believe was on January 10th, we do see this big drop. So there is this correspondence between rainfall in the area and salinity on the reef. So what that means is one thing: we can't necessarily expect to control salinity on this reef simply by decisions we make in terms of discharge. We have unregulated runoff that's going to contribute from the entire area, such that even if we keep all of the locks closed, we can expect small changes in salinity or even moderate changes in salinity to occur as a result of just rainfall alone. But what about freshwater discharge? So you're all familiar with all the regulatory control structures. Curtis talked about these 3, but did not talk about the fact that there's also one here at the mouth, or at the input of C44 into Lake Okeechobee. And so if you look at the discharge rates over time at these different outfalls, a couple different interesting things to note. Number one, during this entire time here, from roughly the end of October to the end of November, when we have very little rainfall and no changes in salinity on the reef, we're actually having water come out of Lake Okeechobee into our canal system. Now, the fact that most of that's coming out of Lake Okeechobee, but then not being reflected in the C44 canal, so notice that red is elevated here but not blue, essentially means that this water is being drawn into the canal and then used or distributed into different areas along the canal rather than going all the way into our estuary. So we have choices about how to use that potential water. The other really interesting thing from this data is that during that high rainfall event, rather than this entire catchment basin running down into C44 canal and then into the lagoon, during that time, all of that water ran into C44 canal, and this lock was open such that water ran backwards into rather than out of Lake Okeechobee. So it's not always the case that we're going from Lake Okeechobee into our estuary, and that's the source of potential problems. We have flexibility and choices in how we handle rainfall in this system. So we are starting to get a better idea of how rainfall and discharge events can impact things like salinity and temperature on the reef, and we know that there is this direct correspondence. Our next question was, how do these discharge events actually affect coral health? And you heard Mr. Gregg talk about quite a bit about linking estuarine health to coral health. And while it's very true that in multiple locations throughout the Caribbean and Florida, there's been a strong link between the health of seagrass systems, mangrove ecosystems, et cetera, on coral reef health, there's essentially been nothing done at all in this region trying to look directly at the effects of this discharge water on coral health. So this was a first look that we started in 2010. And again, we were looking at those same two species, the Pseudodiploria clivosa, or the brain coral, and Montastraea cavernosa, or the star coral. So this is a whole colony shot. Here's a close-up of the two. And as a reminder, they have those algal cells that live throughout their entire tissues. So we wanted to look at 3 response criteria. The first was bacterial communities on the surface of the coral. So in the same way that our entire bodies are lined with bacteria, some of which are beneficial, some are pathogens that are lying in wait for you to become stressed out, corals are the exact same way. They're completely covered with bacteria on their mucosal surfaces, and looking at the kind of bacteria that are there can give you an indication of stress. We also wanted to look at gene expression. So by gene expression, I mean corals turning on genes in response to some kind of stimuli. So in the same way we might turn on genes to make digestive enzymes when we smell a bakery, corals can turn on genes to respond to salinity stress, to respond to temperature stress. And we can look at the individual genes that are being turned on with a number of new approaches. And then lastly, we wanted to look at those zooxanthellae, or the algae that are within the cells, to see how and if they're responding and how they are in comparison to other places in the world. So here's a typical kind of day where we go out and there is a discharge event going on. You can see this sharp demarcation between offshore water and discharge water. We typically see high abundances of reef fish, perhaps even higher than you might expect for an area with the kind of coral cover that we're seeing. St. Lucie Reef is not the best place for corals. We're at the thermal tolerance limit. We're only in the neighborhood of 1 to 2% coral cover, and that's been its baseline for at least 8 to 10 years since this was first looked at. So it's never been the prolific 70% coral cover reef that you might expect in the Southern Caribbean. Ever since 2012, every single time we've gone out, we've seen at least one lionfish, so they're certainly present in the system. We've not yet had an opportunity to evaluate their numbers within our sites. So at each of our locations, we take small coral biopsies of tissue to bring back to the lab to do those 3 different analyses I just mentioned. And essentially every time we go out is a default reef cleanup where we're picking up cast nets off of the reef, usually that are entangled in branching corals. And to give you an idea of scope, essentially for each day we spend on the water, we're on the neighborhood of 3 to 6 weeks of analytical time in order to get the data out of those samples. So it takes some time. Don't worry too much about the left-hand side of this chart. This is just an entire list of all the genes that we used on what's called a microarray. So this microarray is looking at the individual genes in the coral that are turned on. And the colors here are just grouped into different kinds of gene families. And we selected the genes that we wanted to look at. We put 148 different genes from both the corals and the algae onto this microarray, and a total of over 2,000 features or individual little points on the array where we're taking a measurement. The take-home message from all of this work is that we saw expression of stress-related genes elevate during discharge events. And most of those elevated genes were genes that involved cell metabolism, so trying to survive, tissue repair, so there's some kind of tissue damage going on and they're turning on tissue repair genes, and osmotic stress-related genes, or they're experiencing salinity stress of some kind. So this was a really good way for us to be able to tell that, yes, these corals are experiencing stress, even if we can't visually see death or some kind of discoloration or mortality occurring on the corals. So it's a good metric that gives us more detail than just life or death. When we analyze the coral bacterial communities, it's a very large and disparate and difficult kind of data to address. So what I've done here is simply take each individual dot on this graph represents the entire microbial community of a single coral. And so dots, or features that are relatively close to one another on this graph, mean that these two had very similar microbial communities. Dots that are very far away from one another on this graph indicate that they had very different microbial communities. And so we looked at how these microbial communities were changing over time, and there were two big messages to take home from this. Number one, we don't see a big shift in the microbial communities directly after a discharge event. There does seem to be some resilience in the microbial community structure, but not too surprisingly, we do see them change cyclically over time, over a year. So we should expect them to change. We should not expect them to always be the same. And the fact that we might see a large shift is not necessarily related to discharge. It could just be related to temperature, light, etc. But what was very interesting is, again, we've done this same technique in multiple places throughout the Caribbean. So when we compared those microbial assemblages on St. Lucie corals to those from other areas like the Florida Keys or Virgin Islands or Bahamas, we found that they group out completely separately. So here's St. Lucie Reef over here, here's everything else over here. So what that means is that our reefs have very unique microbial assemblages on them relative to other places in the Caribbean, which also indicates that they're likely going to respond differently to environmental changes than other places in the world might. So further evidence that St. Lucie Reef is indeed unique and may require its own set of management criteria. And then lastly, we looked at, again, those zooxanthellae within the cells. So the way we do this is we take a piece of coral that looks like this. We actually use a dental water pick to blast all of the coral tissue off, leaving behind the bare coral skeleton. And then we can then separate out all of the algal cells that come from this tissue and count them. And we scale it to the relative size, so we can get a number of algae per square centimeter or per square inch on that coral fragment. And here in the light bars is the Pseudodiploria, or the brain coral. The dark bars are that star coral, Montastria. And what you'll see here is that one of the corals has many more zooxanthellae per unit area on it than the other coral does, and that's universally across site and over time. So again, these two corals are going to behave differently and respond differently to potential stresses. So tracking just one of them is probably not effective. You need to track both. We wanted to not only look at how many were there, but which types of algae were there. To do that, we did a genotyping analysis. In other words, we fingerprinted these corals to see which kind were there. each of these letters over here corresponds to different types of zooxanthellae, and these can essentially be considered species. So this is an area of research that not enough work has been done to even describe these species, so we're still just bitting them into big groups. There's a B type and a C type, and then there are subtypes within each of those letters. The important thing to take home from this is that we have, again, two completely different types of zooxanthellae within these two different species, indicating, once more, we've got an incredibly unique situation at our reef that does not occur in other places throughout the Caribbean. So, to summarize, we know that canal discharges in rainfall can both impact salinity and water quality issues at St. Lucie Reef. I didn't present much of it today because it's a very even more convoluted analysis than some of what I presented, but we have fairly strong data indicating that rainfall and discharges, both during and during times of upwelling and during times without upwelling, can affect the ability to see the reef, water clarity, for example. And so both of those things we know are going to be impacting our reefs, and we have control at least over one of them. We see significant increases in coral stress gene expression, during the summer discharge events. One of the issues with this, though, is that our discharge events typically occur during the rainy season, during the summer. So the discharge events are also occurring exactly when it's hot out. And so it's impossible in an ecological design like this to tease apart the relative effect of the discharge versus the relative effect of the high heat. So we've actually just started an experiment where we're going to decouple those two, test with discharge water and high temperature, discharge water, low temperature, and the two corresponding opposites, so that we have a controlled experiment where we can look at the effects of discharge water and temperature on coral health, and that's all on site at Harbor Branch. We also know that they harbor these unique microbial assemblages that are completely different from anywhere else in the Caribbean, so may mean they react differently in terms of management, and that's reinforced by the differences we see in zooxanthellae. In terms of our ongoing and future work, I already mentioned those mesocosm experiments. So mesocosms are just small aquaria that are meant to replicate the ecosystem that's out there on the reef. So we're going to be taking corals and exposing them directly to discharge so that we have a more controlled way to measure how they respond to these discharge events. We're also really interested in genotyping the corals themselves to determine which genotypes are resistant to discharge stress. As we move more and more towards active strategies for mitigation and restoration in Florida, in other words, growing corals both in the ocean and in land-based systems to repopulate areas, we have to understand what the role of genotypic diversity plays in who's strong versus who's not, and that might mean putting more effort towards those that we know are resistant, while at the same time recognizing we can't dilute the genetic diversity in the population, because that's risky as well. And then lastly, we've been working towards an enhanced water quality monitoring program both within the estuary and on St. Lucie Reef. So already in place, already in the works as a result of the $2 million legislative appropriation, will be the installation of the 5 new Lobos, which I'm sure you guys are all familiar with. So those are all going to be within the estuary. What we're trying to do is develop a plan to expand that kind of detailed real-time water quality monitoring to the reef itself. So we've just started a project in collaboration with NOAA where we're going to try to develop what that should look like and whether or not St. Lucie is the right place to do it. So basically, we're trying to put those controlled experiments with monitoring and these advanced molecular diagnostics to create better data for more effective management, and I'll be happy to take any questions.
Do any of these species that are on the reefs, do they fall within the designations of threatened, endangered, or no, none of that?
I take it back. Neither of the two that we're working with do. There's one species that is in that group of 8 that was recently added to the endangered species list in their threatened status.
A species that inhabits the reef, our reef.
Correct.
Okay. And so discharges then threaten the existence of that threatened species.
Correct.
Are there other questions for Dr. Voss, please? Commissioner Smith.
Just a thought. So the lobos that you're looking for the reef, what's the status of that? Is that strictly through NOAA, or is that going to be through the legislature, or what?
I think the first step is to get some more lobos installed and demonstrate that they can effectively work to give us the data that we need. The next question is whether or not that same kind of configuration could be suitable for an offshore deployment. And that would involve discussion with Lobo, Saltantic, wet labs, et cetera.
I'm just wondering what we can do to be helpful to help Harbor Branch advance that discussion with the legislature like last year and so on. I mean, merely the conversation is helping, to educate us, but take that in the next step. What do we do next to help that?
I think that there's a clearly recognized need for improved water quality data for these offshore sites, and Curtis alluded to the working group that's been created relative to that. And then this other initiative is essentially a grant from the NOAA Coral Program. The first year is for planning to try to decide where and how to set up a water quality monitoring network. And then year 2 is to implement it, at least it's somewhere within the SEFCRE region, as to be determined. So if the commission wanted to advocate for this location to that group, that would certainly be helpful.
And I think what would make sense, and maybe there's a whole bunch of other data that goes into how you decide, but if you've already been studying 4 sites on the Northern Reef for X amount of years, wouldn't you want to kind of keep the sampling consistent with those sites so you knew additionally what was happening right at those specific areas where you're already doing that?
I would agree with you, but there are certainly, these 4 sites are not the only sites that have been repeatedly sampled within the SEFCRE region.
But these are the most important sites.
Well, that's what I've been trying to tell them on the phone call so far.
So that is something that we could do, and we could.
I think what differentiates these sites in particular is the fact that we will before a decision is even made on where to install this network, we will already be moving towards the installation of the 5 lobos within the estuary. So we certainly have far more information about the upstream water coming to the inlet, and certainly more real-time information than will be available for some of the other locations.
And I know I should know this, but I guess I don't. We have none of the lobos are to be installed in the lagoon. They're just in the sea, in the St. Lucie River, or there's
no. They're throughout the estuary.
You will have them in the lagoon, and we'll be monitoring that as well. There
are currently 7 candidate sites, and we're pretty sure what the 5 are going to be, but we've not made a hard decision on that yet. That's being led by Dr. Dennis Hanasak. I have been advocating quite strongly for one at the weather station right there at the mouth of the inlet. That would be most informative for me. But there are no plans for any of those 5 to be located outside of the inlet.
And so who do we communicate with to support that?
I think that there's 2 possibilities. I think that if we can demonstrate successfully that these 5 lobos are the way to go, then that would justify an additional legislative request to expand that kind of approach outside of the lagoon. And I think there's other avenues through partnerships with NOAA, DEP, et cetera, that could also be explored.
Got it. Fantastic.
Others with questions, please.
I'm sorry, sure. One more thing. What would be helpful, I think, from Harbor Branch, and NOAA, if that's appropriate, is to put a 1-page white paper or something together that we had that would be a take-along, something that we could talk to our legislators about, even in anticipation. I mean, to sit through, and no offense, but to sit through the length of this, I don't know if our legislators will take as much time that has been done here. So it might be helpful to condense the ask or the strategy of the ask, in a 1-page something that we could take with us and say, here's what we're looking for help on this coming session or next year.
That's something I would be very happy to provide.
Great, thank you.
That's most kind, yes.
Very enlightening work. Thank you very much.
Mr. Scott, did you have something you wish to say? If there's none other, we'll bid you adieu and tell you both thank you so much for what you've done and what we anticipate you're doing and our encouraging you to accomplish. I tried to say that tactfully rather than extract from you. So,
Thank you all very much for your time. I appreciate it.
Thank you.